Okay, thanks very much. I'm Sean Ainsworth, CEO of Immusoft. We are developing B cells as a novel modality for therapeutic protein delivery. More specifically, we're working with plasma cells, which are a subset of B cells that are extraordinarily prolific at protein production, which is why we've selected those as a modality for protein delivery. And so naturally, B cells are producing and releasing thousands of antibodies per second into the bloodstream. And so initially, we started interrogating those and programming them, if you will, to produce bespoke proteins, bespoke antibodies, in particular against HIV, and began to ask the question as to whether we could actually get them to produce other protein types, and started working with things like enzymes and signaling proteins, and found out pretty quickly that plasma cells, under the right conditions, are quite versatile and able to produce a number of different therapeutic proteins. And so we've been working on that as a platform. I'm sure you can appreciate under each of those different protein types there are verticals with each with tremendous potential opportunities. We are initially focused on MPS I as our first-in-human approach. And so MPS I, it's a lysosomal storage disease. We've got tremendous data around that in the way of preclinical safety and efficacy, and we expect to be in the clinic, or at least to open IND later this year and into the clinic shortly thereafter. I guess hot off the press here this morning, we announced a deal with Takeda that we're very excited about. We're collaborating with them on the use of B cells as a modality to cross the blood-brain barrier, and in this case, address, in particular, neurometabolic indications. So the deal is valued at up to $900 million, or actually over $900 million if you include all the milestones and bells and whistles. So Takeda, we think, is just a tremendous partner with deep expertise in neurometabolic indications, orphan diseases, and a very deep commitment in advanced cell and gene therapies. One of the main things that I really appreciated in coming over to Immusoft was the ability to evade what I think are some of the major issues associated with current modalities of gene delivery. In particular, if we think about systemic virus delivery, we avoid the immunogenicity issues associated with viruses. And in the way of gene-modified stem cells, we don't require the busulfan regimen that's associated with those stem cell approaches. And when we think about enzyme replacement therapies, while it's not advanced therapeutics per se, we think that this is actually a pretty interesting market and potential opportunity for us, where we can address what are some substantial unmet needs, namely the need for these patients to be dosed every week, 4 hours at a time with their infusions, and the sawtooth effect that comes along with weekly dosing as is done. And so the way this works, we start out with an apheresis. We isolate out the B cells from the apheresis product. And then in our culture system, we introduce our therapeutic gene of interest. We use a sleeping beauty transposon system to do that. We then expand those B cells, differentiate them into plasmablasts, plasma cells, which then get dosed back to the patient, where we expect durable expression. We know that long-lived plasma cells have a half-life of roughly 17 years, and so we expect many years of durable expression from our approach. That having been said, we do anticipate that we will be able to redose these patients because this is non-viral. There is no need for any type of preconditioning regimen. And so if we isolate in on the Sleeping Beauty transposon system, there are really a number of advantages of this over virus-mediated approaches. So namely, we avoid the immunogenicity associated with viruses. We have the ability to deliver much greater, larger payloads. If we think about the CMC, it's dramatically simplified. Plasmids are much more reproducible, stable than virus. Not to mention less expensive, shorter lead times. So a number of real advantages with this system. And last but not least, we've got a pretty substantial body of preclinical safety with this. And there are others that have taken it into the clinic in a T-cell approach and demonstrated both preclinical and clinical safety around the transposon system. And so we started out with the B-cell approach, initially looking at antibodies as a modality of protein delivery, and moved from there into enzymes. We've delivered also signaling proteins like follistatin and other therapeutic proteins. And so if we think about each of these different protein types with enzymes, we can pursue lysosomal storage diseases, antibodies. We can get into things like autoimmune, oncology, infectious disease, and so forth. And so really there's a great deal of opportunity out there for us. And so if we look at just one vertical, the enzyme replacement approach, we start out with MPS I. And what we anticipate here is that if we show proof of concept, as we anticipate we will, then we can start to move down this list. And the beauty of this vertical is that these are all proteins that are currently approved. They're in the clinic, or they're on the market. And but nonetheless, they have we think pretty substantial unmet need and would benefit from the 24/7 delivery that the plasma cell approach would do. And so looking at our pipeline, we are starting out with Hurler syndrome and moving relatively quickly thereafter into Hunter syndrome, MPS II, which dovetails very nicely with our lead program. We've got some great data as well with follistatin protein, which is a muscle-building protein, if you will, and are contemplating ALS, DMD with the follistatin approach. And from there, we'll look to go into additional lysosomal storage diseases and then into cardiovascular, autoimmune. And as we've noted here, we're proud to be partnered with Takeda in the CNS realm. And so a little bit about mucopolysaccharidosis. It's characterized by a deficient or absent iduronidase enzyme. And that enzyme is involved in the breakdown of glycosaminoglycans. And so when those glycosaminoglycans build up, they are deleterious across numerous tissue types, organs, and organ systems. And ultimately, they will, if untreated, the severe forms of this disease those patients will have a lifespan of roughly 4 years. Thankfully, there are treatments on the market with Enzyme and with bone marrow transplant, but there is still unmet need in this arena. And so these patients live into their 20s, but at present, not really too far beyond that. And so what we are hearing from the clinicians that treat these patients is that more enzyme is better. And so there's a constant drive to find ways to get more enzyme to these patients. And so we anticipate that the 24/7 delivery that we'll be able to effect in this patient population will overcome some of this unmet need. And so just a quick look at our data. We've got, this is one snippet from numerous studies that we've conducted, but what all those studies have demonstrated is that we show an increase in the iduronidase enzyme in the plasma, and that's this the horizontal red bar. Those are supranormal levels of iduronidase, and we showed decreases in glycosaminoglycans across numerous tissue types. We're currently expecting to get to the open IND here this quarter before the end of the year, and ultimately be in the clinic. This is for MPS I program, ultimately be in the clinic shortly thereafter. We do have orphan disease designation and rare pediatric disease designation for MPS I, and we are actively working on our MPS II program and excited to be bringing that forward into more fulsome development real time. So this is just some pilot data from our MPS II program. And what we see here is that the urinary gags in the middle here are reduced nearly to the level of the wild-type animals, and particularly where we saw good engraftment in those animals. Those 3 of the 4 showed very dramatic reductions in glycosaminoglycans. And the data that we're seeing in MPS II dovetails very nicely and tracks very closely with what we're seeing in MPS I. So as far as manufacturing goes, this is an area that we've put a tremendous amount of resources into. We've had an ongoing dialogue with the FDA. We've done now numerous PDE and engineering runs and showed great reproducibility across those runs. We've got all of our GMP source materials. All of our assays are set and ready to go. And so we're very confident in our ability to reproducibly manufacture our cells. This is really a tremendous management team that I'm proud to be working with. And Rob Hayes, he was formerly head of global biologics for Amgen. Excuse me, Bob Sikorsky, former CMO at 5 Prime Therapeutics, a long track record of success in the clinical arena. Cam Gallagher, as a serial entrepreneur, had 2 huge exits last year, and I actually worked with Cam previously at Retrosense Therapeutics, which we successfully exited to Allergan. And Monika Svidlicka was instrumental in the development of the Dendrion program, which was ultimately the first human cell therapy approved by the FDA. So a little bit about our intellectual property. We started with IP out of David Baltimore's lab at Caltech, and that was around the production of antibodies to HIV using plasma cells. We've since augmented that with intellectual property from University of Minnesota, Max Delbruck, Discovery Genomics Inc., around the Sleeping Beauty transposon system. And we have continued to build on that with in-house intellectual property filings. And so we've got really the dominant IP position in this space, which I would define as the use or manufacture of B cells as therapeutic modalities. We've got freedom to operate as well, and we expect many more years of IP coverage on our current portfolio. And so to summarize, this is a new innovative platform that we are developing using B cells as modalities of therapeutic protein delivery. We overcome what I think are some of the unmet needs with respect to other gene delivery modalities, namely immunogenicity associated with viruses, the need for preconditioning associated with stem cell modalities. It's a huge market opportunity here. We've successfully delivered a number of different protein types. We're very excited about being in the clinic here with our lead program in the not-too-distant future, and likewise tremendously excited about our relationship with Takeda in neurometabolic conditions. And we're looking at that relationship as kind of a blueprint for some of the other discussions that we're having. And if anyone's interested in learning more, I would be happy to talk to you about that. Thank you very much.